Um, Thank you, first of all, to the organizers, Ruben and Mary Lou, and everyone who got me here, and to Randy for organizing this session. Today I'm going to talk about a very different kind of situation that I've been involved in. And the work that I'm going to be talking about today is primarily, primarily that of Matt Casson, who is a PhD student who graduated just last year from our department at Penn State. He's now a postdoc at Virginia Tech working on thousand canker, of all things. And Matt is a force of nature who took on this project on Fusarium, collaborating with me in association with his disease, which is verticillium wilt of Ailanthus, a Fusarium ambrosia beetle story. That I think, it's going to be different from what we've talked about otherwise, but hopefully we'll be able to make some connections that'll be useful for food for thought. Matt worked with my PhD student, Dylan Short, on the molecular and Fusarium aspects here. He's now a postdoc at UC Davis working on verticillium, of all things, with Krishna Subbarao. And Christy Fenstermacher is a current new student in my lab at Penn State who's been kind of finishing up on this and might actually incorporate a significant ambrosia beetle story in her project as well. There we go. So I feel a little bit, though, like if you remember Admiral Stockdale, Ross Perot's vice presidential candidate in 1992, who am I? Why am I here? And I'm hearing, and it's been fantastic hearing all of this. I've learned more about ambrosia beetles in the last day and a half than I have in my entire life. But I do ask, why am I here? And I do want to make sure that I make it clear that, I'm here to help. First, in addition to my own interest in these stories, I want to emphasize, helping in terms of Fusarium systematics. And I think Carrie, the same thing can be said. I'll speak for Carrie and say that he feels the same way. We're here to help with Fusarium systematics. Cultures between Carrie and myself, we've got almost 40,000. We've got culture collections with almost 40,000 accessions. And so we've got great culture collection resources and for basic Fusarium biology. So please ask if you've got questions and want help. Whoops, I'm going the wrong way. And I just want to put in one plug. There is a USDA-sponsored systematic internship program that I'm involved in for a summer intern. So if you have a student, technician, somebody who really needs to spend a summer working with Fusarium in kind of a systematic context, contact this website here at the University of Florida. It's Amanda Hodges is the one who's running this program. There's also Anthony Cognato is one of the mentors for this at Michigan State, who is a beetle guy. So that's another option. So if you need somebody who needs some systematics experience for a summer, this is an opportunity for doing that. Keep going the wrong way. So ambrosia beetles, symbiotic fungi, and tree diseases. And what I've learned so far this week, there should be a label. Oh, there it is. Here, I've learned about dieback of avocado and other hosts in Israel and California, where you've got Eualacia fornicatus, which might be a species complex, and the pathogen being an undescribed Fusarium species in the Fusarium ambrosium clade. Is there a Raffaella that's involved here? Is there one that's present? I'm gathering not. I haven't heard anything about one. And then you've got red bay laurel wilt, where you've got Xyloborus glabratus as the insect pest / vector. The pathogen is a Raffaella, not a symbiotic Fusarium in that one, I think. Maybe people can correct me if I'm wrong. Well, and that's a big threat to avocado there. Well, I'm going to talk about a very different story. This is verticillium wilt of Ailanthus, where there's an insect that might be a vector, an ambrosia beetle, Eualacia validus, which was one of the things that Bob Rebellia talked about, where it was not on his list of important ambrosia beetles. And the pathogen is a verticillium non-alfalfa. But the symbionts are there. It's another undescribed species in the Fusarium ambrosium clade. And there's also an undescribed Raffaella species. So those are familiar aspects here. So you might be asking, if the beetle and its symbionts aren't killing the trees, who cares? Well, I think it's important, let me go back here, to understand a situation gone wrong, which I think is maybe what we're talking about with these serious tree syndromes. It's useful to understand what's normal first. And I think that the verticillium wilt ambrosia beetle story is kind of what's normal for these things. And so I offer this as one example. So we've got, and I messed this up, one beetle and one fungus appeared to be involved, but the symbionts with the beetle are not involved in the disease syndrome, if it is a disease. In this case, it is. Now, there are other things to consider in terms of what might be going on. These fungi and their hosts, and I should have said this differently, there could be hybridization going on among different members of the Fusarium ambrosium clade, and there may be hybridization going on among the different ambrosia beetles, which is a complicating factor. The hosts may just be exchanging fungal symbionts over time. And then, and finally, and we've seen some evidence for this in Carrie's talk, the fungal symbionts may actually interbreed. So I'm talking about Ailanthus altissima, which this is a picture I just took yesterday of the big, beautiful tree of heaven in front of the entrance of the Huntington Gardens. So there's the tree of heaven, and then what is referred to sometimes as the ghetto palm, which is the invasive tree that you see on roadsides, in urban landscapes, where in this case, I found this picture of one growing between two brick walls, between two buildings, and they tore down the one brick wall, and here's the tree still alive, still going strong after that. So it's a very serious invasive tree that people would like to kill. And it was also the book A Tree Grows in Brooklyn. That's actually Ailanthus here, a tree of heaven, the famous tree that grows in Brooklyn. So verticillium wilt on Ailanthus. This was discovered in about 2005 in a stand of otherwise mixed hardwood coniferous forests in south central Pennsylvania. Ailanthus established there after an extensive episode of moth deforestation, and then they salvaged removing all of the dead trees, and then Ailanthus came in the 1970s. And so this Ailanthus, in this picture, you can see that there's all these dead trees. This established probably starting in the early 1980s. So these were all killed by verticillium in this patch. Since then, it's been found in multiple foci in Pennsylvania and surrounding states. The pathogen was first identified in Don Davis's lab as verticillium alboatrum, but later refined as verticillium non-alfalfa. And there's a lot of interest in this because people want to kill verticillium potentially as a biological control agent. So now the next story is Eualacia validus on Ailanthus. And so Matt looked at 4 sites in Pennsylvania averaging about 66 kilometers apart. And first of all, he found that there was large numbers of these ambrosia beetles at his original site. And then he actually inoculated trees in additional sites after finding that. So he looked at this naturally occurring site pictured here, as well as 3 other sites where he inoculated 25 trees with their known verticillium pathogen. And he found that Eualacia validus infestations began at each site within weeks after wilt symptoms appear. So this is a very fast-acting pathogen. It only takes a few weeks to get the symptoms. And then a few weeks after that, boom, the insects show up. So all of the inoculated trees were heavily infested within 1 year. And then you would see spread of the wilt, and you would also see spread of the occurrence of the insects as well over time. So here's just some pictures of some trees. And what Matt would do is bring in, cut bolts from these, and then collect beetles, and they're just all completely full of these ambrosia beetles. And I actually use this in my mycology course as an isolation project because we can reliably isolate a few interesting fungi in a mycology class without all of the weed fungi like cladosporiums and penicillium species that really give mycology students a bad experience when they're trying to identify them. These are a little bit more straightforward. Eualacia validus is an introduced species, originally from Japan, East Asia, and was previously known as Xyloborus. It has a wide host range in Asia, but Bob Rebellia can correct me where I'm wrong, only on dying or dead hosts. And it was first detected in the US on Long Island, New York in 1976, and now it's fairly widespread in the mid-Atlantic. And I believe that there was a couple of southern states maybe where it's been found as well. And it's known in Pennsylvania anyway, on Ailanthus, black oak, and yellow poplar. We don't have it. It hasn't done anything to our avocado industry in Pennsylvania. It's gone from 0 to 0, so it stayed the same. So here's Eulacea validus in this tree from the Cognato et al. And you can see that it's a fairly close relative to Eulacea fornicatus. Xyloborus glabratus is here. Whoops, keep going the wrong way. And the first thing that Matt looked at was, were mycangia. So these insects do have submandibular mycangia, and Matt did cross sections and found very nice mycangia with fungal bodies contained within them. Can't tell if this is a Fusarium here or a Raphaela or both all mixed together, which is kind of interesting if they pack together like that, the two very different fungi. Oops. Nope, I'm going the wrong way again. Maybe if I do it this way. So what Matt found was that these fungi are indeed associated with the head. And you can see, wait a minute, I do need to do it this way. This is very different from the one that I'm used to. So looking at Fusarium species, and we've got our Raphaela species. And again, these are both undescribed. And then sometimes there's a graphium-like thing that we would find. Fairly abundant in heads, both fungi. And there are other fungi that you can isolate as well. So these are dissected heads, surface sterilized. But really not much in the thorax. No Fusarium, no Raphaela in the thorax, and none in the abdomen. And so these are things that look, when you're doing plating, very different from each other. You've got colony 1 and colony 2. One is the Fusarium, and you can see this pretty typical Fusarium ambrosium-like morphology, these macroconidia here. And then also, here's the Raphaela species. Now it's not responding at all. There we go. In terms of life stages, Matt isolated from various life stages. You can see that the Fusarium species and there's a graphium as well, as you find it quite a bit in the larvae, and almost nothing in the pupae except for the graphium and a few other fungi. You see a little bit of Fusarium in the adult males, but not a lot of fungi, but it's the immature and adult females where you find both the Fusarium and the Raphaela. So he only found the Raphaela in the adults, didn't find it in larvae, but he did find the Fusarium in the larvae. So there's one interesting difference. So Matt, being interested in this particular disease, did inoculation studies asking the question, is the Fusarium or the Raphaela involved? And so he inoculated ailanthus, tulip, poplar, and black oaks, trees that were also co-occurring in these stands with the ailanthus. And so he bored holes in trees at a particular height, cut out little auger plugs from cultures, put them in there, and then sealed them over. And he found no disease caused by, compared to controls, for either the Raphaela or for the Fusarium. Similar experiments with the verticillium, though, take care of it. It definitely is a pretty potent pathogen. Okay. And so this is just a picture of that, showing that this is what sort of a negative reaction looked like compared to controls. So what is the role of the insect in this disease? Well, we really don't know. It could be that the ambrosia beetle has a role in passive transport of the verticillium. You can isolate verticillium non-alfalfa from beetles that are not surface sterilized, so it does appear to be on their bodies. And beetles can be found on healthy trees where disease and infestations are established. But you don't tend to find beetles around where the trees are not diseased. So they come in once the disease is there, and that's the only place where they're seen. And again, I think that this is very typical of an ambrosia beetle story. So Matt also did a phylogenetic analysis of Fusarium species from Eulacea, and Carrie essentially told this story before. I'll just point out a few things. Again, the ambrosia fusaria represent a strongly supported monophyletic lineage, shown here as lineage A in the Fusarium solani species complex. And here I've got represented some of the isolates from Israel, from avocado. Here are the ailanthus, Eulacea validus strains, and they're all clonal at all of the loci. And here we've got ambrosium from T, and here's rubber tree, Fusarium ambrosium. Now, Matt, so all of the Ailanthus, E. validus associated Fusaria share the same multi-locus haplotype, and they appear to be closely related to the Israel avocado type, but also to the T type. All known Fusaria from, one important point I want to make is that all of the known Fusaria in this clade A, they either came from an ambrosia beetle or they came from galleries of an ambrosia beetle. Now, maybe if we look long enough and hard enough, we'll find them from other niches. But right now, this clade appears to be very tightly co-evolved with ambrosia beetles. So that's one important point I want to make. The other important point I want to make, and so this is just a close-up of that, is that all of the known Fusaria in the Fusarium ambrosium clade produce a morphology that's really very different from what you otherwise see in the Fusarium solani species complex. This is typical Fusarium solani macroconidial morphology, those canoe-shaped or banana-shaped macroconidia. You don't see that in Fusarium ambrosium. They have these irregularly wide sort of clavate conidia. Now, in this neighboring clade, clade B, and you can see that there is one isolate here from X. ferrugineus. So there are some things associated with ambrosia beetles, but also from a number of other hosts, including the parathecial isolate from Sri Lanka that Carrie mentioned. At least in terms of this isolate from French Guiana and the Costa Rican X. ferrugineus isolate and the dead tree Sri Lanka isolate, these have a much more typical Fusarium solani morphology. And my hypothesis is that there's been some really tight coevolution here on this lineage A that has led to a very strong association with the beetles, and there's morphological and probably a lot of other traits that have gone along with that. So I think it's a really interesting group. So again, members of that neighboring clade produce the more typical Fusarium solani type morphology. I also show again here that we've got these two isolates from T that share their elongation factor 1 alpha allele with Ailanthus. So just to bring up this subject again, that there is potential intragression or interbreeding, and I would probably predict interbreeding, but it could be something akin to horizontal gene transfer, which is known in the Fusarium solani species complex in the sense that their genomes contain a lot of genes that nobody knows where they came from, if that is a sign of horizontal gene transfer. But it's an interesting thing that we really need to look out for is genetic exchange between these different ambrosia beetle associates. And I'm going to finish up. So just in conclusion, Eulacia validus on Ailanthus has two unique mycangial fungi, a Fusarium species closely related to Fusarium ambrosium, and a Raffaella species that hasn't been characterized further, but based on on ITS data, looks quite different from anything else that's in the databases. Fusarium isolates from E. validus represent a monophyletic, apparently clonal lineage closely related to ambrosium, but we've got some evidence for genetic exchange here. The mycangial symbionts from at least two Eulacia form a strongly supported monophyletic group within the complex. And tree inoculations, though, with the Fusarium species from E. validus reveal that both the Fusarium and also the Raffaella are not pathogenic, or either on Ailanthus or on other species in Pennsylvania forests. We don't know if it's pathogenic on avocado, though. So with that, I'll stop, and if there's time for questions, answer them.
So how many loci do you look at when you use your multi-locus sequence typing?
Well, our classic Solani complex typing, it was RPB2, elongation factor 1 alpha, and one piece of the ribosomal DNA that includes the ITS in the large subunit. Carrie has recently added an RPB1 data set as well, which is highly informative. Okay. So, but you can do a pretty good job with the first.
Yeah, because your ability to detect this recombination stuff depends, of course, on how many loci you're looking at.
Absolutely, and that's a very important point to consider. And I think, as Carrie said, we've really scratched the surface here in terms of the diversity. Yeah. How many beetles are there in this clade? And it may be that that we'll find, is this going to be, we're going to see these examples all the time, or did we just happen to find the needle in the haystack with this one elongation factor allele? I'll let him.
David, you mentioned that you isolated the graphium species from the larvae. So have you done any pathogenicity tests with that, whether it is pathogen or not?
No, Matt did not do much with that. I'd be curious to know, though, if there are any reports of things like that having any such role. I'm not aware of it myself. Yes?
I loved your graph when you showed that there is a whole bunch of Rafflesia too in the system, and that I've mentioned something like that to a bunch of people here. I'm curious if we have Rafflesia and Fornicatus too, and so the reason we might not see it is because, in my experience, Fusarium has many more spores than Rafflesia, and I'm wondering about your opinion on when we count colony-forming units. What is it that we are actually counting when it comes to those Rafflesias that, they're mostly really mycelium, as opposed to Fusarium, which produces a lot of spores?
Well, we don't know, right? I mean, you're plating out what you're plating out, and there's sampling error potentially there. But then on top of that, of course, growth rates vary. So I would strongly recommend that actually for any given insect to do an experiment first with definitely with different dilution schemes, but potentially also with different media to see if you're getting different results using different methods. And maybe it's time to start doing metagenomics too on top of that, some sequence-based stuff, and seeing if you get the same results using different methods.
I was just wondering with the two fungal symbionts whether they could be acting synergistically with each other, or whether they're occupying different micro niches within the brood gallery, sort of different time periods of appearance and stuff?
Well, I think that's a really interesting question, and I'd really like to hear what people who have a lot of experience looking at these beetles and others that have fungal symbionts see. At this point, I don't know the answer to that, but the fact that they do co-occur, and again, in the balls of fungus in those mycangia, do we really have Fusarium and Rafflesia stuck together in a dough ball or whatever it is? Because that's not something that fungi readily want to do with one another. So I think that it's a really interesting question, and I don't know the answer.
One more question, we need to go to the next one.
Just as to comment on kind of the coexistence of symbionts, you do see with a lot of these species and bark and ambrosia beetles that there are actually often two nutritional mutualists. And it is weird because fungi usually battle, and a lot of these can get along. They grow together, and it's very bizarre. And they also do seem to have very different environmental tolerances. And so some will grow when it's cooler, some will grow when it's warmer, and it almost kind of adds to the ecological amplitude. We think of some of these beetles by having symbionts that can exist across a broader environmental range of conditions. And we've actually tested that now with mountain pine beetle, and that appears to be the case. It's showing up again with southern pine beetle. We have good evidence in western pine beetle that by having multiple symbionts with different tolerances, you actually do have a broader ecological amplitude. But they get along, which is weird.
Thank you, David.